Well, they are sharing slides. Let me introduce our last speaker this morning. Professor Robert Green from Harvard Medical School, and his title is The Case for Universal Newborn Screening.
All right, great. You can hear me okay? I think I'll walk around, keep you awake before lunchtime. So now for something completely different. How many of you have children? Okay. How many of you plan to have children in the future? How many of you were ever children yourselves? Okay, so basically everybody. So I'm going to sort of talk a little bit, as Les did, about something where biomarkers are going, genetic biomarkers are going, that I believe is expanding, even exploding right now. And I hope you'll agree with me at the end of this talk. So the case is for universal newborn and childhood sequencing, the idea that every single child in the world should have this for them. As we always do, here are my support and disclosures. I'm mostly supported by NIH. Thank you for your tax dollars. Some other foundations, Department of Defense. I advise a couple of companies, and I've co-founded 2 companies, one of which is Nurture Genomics that I'm going to talk about at the end, which is attempting to commercialize newborn and childhood sequencing. So Les and I are medical geneticists, and the bread and butter of what we do today has to do with the diagnosis of rare conditions, a little bit of preconception screening. You got a family who you know has BRCA1, you got to track that down. If you got a metabolic abnormality, you might follow up with some newborn sequencing on a very limited amount. If you're pregnant, you're probably checking for aneuploidy today with the mother's blood. And of course, there's an enormous amount, I've been hearing at this meeting, about targeted therapies for cancer using genetic markers. But what we're also hearing about, and what we know is happening, are these aspects of population screening, the notion that we could find out something important about everybody, and newborn sequencing is a portion of that, along with polygenic risk scores, prenatal sequencing, the idea we could do an entire sequence on a fetus, broadly expanded carrier screening, and of course, the kind of thing you guys spend all your lives doing, expanded and more detailed pharmacogenomics. And where we're heading for lay audiences, this is old hat for you, is gene and cell therapies and integration of the multiple omics, big data pathways that include the exposome, the metabolome, the epigenome, transcriptome, along with genomics. But what's ready for medical practice today, and what's on the cutting edge of that? And I've been fortunate to work in a group that we call Genomes to People, with lots of really great collaborators over the last decade or so, including prominently the ones you see pictured there. And our focus has been on the benefits, the risks, and the harms of using genetics in day-to-day medicine. It's as simple as that. It's implementation science. It's the way less sexy cousin of all the cool basic science that you do. But once you develop these things, aside from therapies in pharma, how do we get them into practice? And so there's a whole science of implementation that we've been working on. Now, you heard in Les's talk yesterday about the groundbreaking work that I believe we did together and with our working group on incidental, or now called secondary, genomic findings back in 2013. And Les is very modest about that, but I won't be. This is the only screening panel for genomics that has ever been endorsed by a professional body in the world in the last 13 years. So it's cracked open the door. Yes, it started with secondary findings. You sequence Matt here for his heart. What should you look for in terms of a cancer predisposition? But as Les pointed out, it's now being used in the All of Us study, the version 2 version, in lots of IRBs around the country and the world, and has kind of propped open that door for what you might consider to be opportunistic screening, but it's the first step toward population screening. And as Les also talked about, there's genomic research findings. If you had to guess, how many people in the world do you think have had at least genome-wide association studies and maybe sequencing all together? Just a broad guess. I tried to do this with some friends at Broad. We came up with roughly 100 million. 100 million people have had either their genome sequenced or at least their array-based testing done. And what percentage of that 100 million people do you think has gotten anything back from that research study? Anything that could help them, anything that could avoid a malignancy or heart disease or something else? It's a trivial percentage. So giving people back, as he pointed out, giving people back information when they participate in research is a really critical issue. And this is just one of many papers we worked on. We are actually giving back genomic results in the Framingham Heart Study, the All-African-American Jackson Heart Study, many different biobanks, both in the US and around the world. And where all this is leading to is if you want to get sequenced, you should be able to get sequenced. If you don't want to, that's okay too. But if you want to, you should be able to get sequenced. So how many people in this audience have not just had like 23andMe array-based testing, but have actually had whole exome or whole genome sequencing on yourself? Okay, I would say that's sad. You guys are in genomics, for crying out loud. Aren't you curious? Don't you want to know? Don't you want to be able to talk about the personal experience of exploring your own genome? I mean, I do this with the residents now and ask the same question, and most of them haven't done anything, and they seem to have no interest in it. I can't even understand what's wrong with them. This is the field they're going into in clinical genetics, and they still have so little curiosity. And that's this notion that somehow the information is dangerous or off-putting, or we have to prioritize this right not to know over the right to find something that might be life-saving. That's insane. So there's tons going on in the area of elective genomic screening and population screening. One such study we did in 2014-15 was called the MedSeq Project. It was part of the CSER consortium. And we found that there is a surprising amount. Now, Les actually talked about the incidental findings with a limited number of genes, say 59 or 81. And he calculated that a couple of percentile of the audience sitting in here. Let me take that one step further. What you find depends on how many genes you look at. I mean, this is intuitively clear, right? But look at it the way we lay it out. If you're just going to look at 10 genes, you're still going to find 1 to 2% of people who are carrying a pathogenic variant in a treatable genetic condition. If you go to the version 2, you get about 2 to 3%. If you go to version 3, you get about 3 to 4%. One of the most popular screening panels happened to be made by Invitae, was 167 genes. You get up to about 6%. And in the studies that we did, the NIH studies we did, we looked at 5,000 genes on average, and an incredible 16 to 20% are carrying a pathogenic mutation in a Mendelian disease, either a monogenic dominant or a biallelic recessive. And in fact, if you look across this, you've got up to 20%. Almost all of us carry recessive mutations, which surely we should know about before we have children. 80% of us with atypical responses to medications. And depending on how many polygenic risk scores you look at and where you draw the threshold for reporting something as high, you can easily get to a point where there's 40, 50, 60% of us who are elevated risk on at least one polygenic risk score. So if anybody tells you that there's not valuable information in your genome, I beg to differ. We just aren't using it. And we've had this notion, it's not a new idea. We've had this notion that we should be using genomic information, ultimately multi-omic information, but at least genomic information at every stage of life, right? From childhood, adolescence, reproduction, middle age, aging, polygenics. pharmacogenomics at every stage. So let me zero in on the youth, because the idea that we'd somehow do this on young children has been around for quite a while. I found quotes back to the early 1990s. You can see there Craig Venter in 2000, Francis Collins in 2009, Alan Guttmacher very explicitly, every newborn will have their genome sequenced at birth. And over the COVID shutdown, I got to know my COVID buddy was Wally Gilbert, who actually sequenced the very first DNA of anybody before Sanger. And when I talked to him about this, turns out that they were talking about this even earlier. So this is like the oldest of cool ideas that 50 years ago, they've been talking about sequencing your children and your grandchildren. And why has it not happened? Well, there's still an issue of cost, but primarily it is because these are individually rare conditions. Even though they're in the aggregate, they're not rare. They individually have a low prior probability. So you are going to identify many children who are at risk, but who do not ultimately develop the disease. This is not a bad thing. This should not be called a false positive, which is the language everyone is using from the newborn screening ecosystem. Because the newborn screening ecosystem focuses on a specific unit of time in the perinatal region. If you're imagining that you're looking at risk over a lifetime, then you use the terminology at risk. You don't use the terminology of false positive, because you don't know if it's false until you follow the child into whatever frame of reference they might develop the disease. The clinical utility and cost effectiveness at scale certainly haven't been proven yet, although the individual clinical utility of discovering and understanding a child and avoiding a diagnostic odyssey is sort of undeniable. And we always come back to this inadequate expertise in the workforce. But in your world, you're creating all these gene and cell therapies. There's 700 or so now, and they are, as I understand it, there's about $1 trillion of investment in gene and cell therapies for rare diseases. So who are you going to call when you've made this drug and you want to use it? Do you want to wait until the child is devastated, or do you want to find them early? And, you know, in case this is unfamiliar to you and just want a sort of few examples, you're thinking, what are you talking about? OTC deficiency, all you really need to do is know that this child should not have a high protein diet, or when faced with a viral infection may get triggered into hyperammonemia. For retinoblastoma, we're still looking in a 18th century way for a white reflex in the back of the child's eye, instead of knowing which children are at risk by virtue of a mutation. Hemophilia, glycogen storage diseases, certain vitamin-dependent epilepsies, and the whole arena of diseases, again, that on average take 5 years of suffering in a family to get to the point where they see the right specialist and get the right tests to diagnose. So I want to tell you the rest of the time about the BabySeq project, which is the project we started in 2013. It, again, has a large number of investigators, over 100, but these are the leaders in the different groups. Our current BabySeq has 3 sites, Boston, Mount Sinai, and Alabama. And BabySeq started off simply asking how many of parents want this for their children. So I'll ask you, how many of you who have children or imagine having children would like, let's set cost aside, would like to get your child sequenced for treatable genetic conditions? Okay. I want to talk to all of you who didn't raise your hands afterwards. Turns out that about 70% of, however you ask this question, adults, children, biobank return, it's always about 70% of people want it if you specify that it's treatable. Then you got to decide which genes should be screened. Now, Les and I talked to you about these adult onset secondary finding panel, which is really appropriate for adults. But for children, there's a whole vast array of genes. So we went through this whole exercise, which you can read about, and we published in 2017, where we tried to make sure the disease gene validity was good, the age of onset was appropriate, the penetrance was on the high side. We came up with about 1,000 genes, and we've since expanded that. So we in BabySeq are looking for 4,299 genes. We're actually looking for genes regardless of whether you consider them treatable or not, because what's not treatable today will surely have a chance of being treatable tomorrow. So we wanted to cast a broad net here. And when we do this, we come up with genes in every medical specialty. Interestingly, just by sheer numbers of genes, neurology is the leader with metabolism, and you can see all the other breakthroughs there. Now, which variants should be reported? I think you're familiar with the notion that there's pathogenic, likely pathogenic, and variants of uncertain significance. But imagine this three-dimensional search space where for indication-based purposes, we'll look for VUSs or variants of uncertain significance. But for screening purposes, we don't need to because the prior probabilities are so low, and historically VUSs tend to get reclassified as benign. So let's just leave them out since we want to minimize sensitivity here and maximize specificity. So instead, we'll go to definitive and strong disease gene validity, pathogenic and likely pathogenic, and of course we'll lean into clinical actionability because that's what we can do something about, and that's what most people want to know. So it's a smaller search space. Now, we were faced with a lot of skepticism when we started BabySeq. It was the first in the world to do this. There were literally people standing up, and this has happened to us a couple of times, right, Les? There were literally people standing up at meetings and committees and yelling at us that this was unethical. So we started BabySeq as a randomized clinical trial to try to be as close to equipoise as possible. And you haven't really felt the heat of this until you've volunteered. You've gotten about a couple hundred families to volunteer, and then you tell 50% of them, no, you can't have the sequence that you volunteered for. That really pisses them off. But nevertheless, we did that, and we looked very carefully at medical, economic, and psychosocial outcomes. And what we found was kind of astounding. What we found was first, the first infant we did successfully screen was on May 22nd, 2015. And this was a pretty exciting day for us. As best we know, there might be others, please tell us if you know, this was the first infant screened as part of a research protocol in the world. And the initial yield on the first 160 or so that were screened was 11%. And you can see some of the findings there. And to our surprise, it wasn't just predicting stuff in the future. It was finding clinical features of a disease that the pediatricians had missed or misdiagnosed. This is not just a window into the future. This is a window into ongoing disease now that has not been appropriately appreciated. Parents were not distressed. This question has been asked and answered in many venues, but is always raised. And this randomized clinical trial methodology really allowed us to dial down into validated scales of depression, anxiety, self-blame, and parent-child bonding. And the economics of downstream spending, there's been a lot of talk about spending the medical commons and how we're going to break the bank. This is definitely going to cost money, and it's going to cost money to follow up. But it turns out that if you compare the controls in that same age group to the children who were sequenced, there's not a tremendous difference in the spending. And even if you look at the children in whom there was an MDR or monogenic disease risk, there was not a tremendous difference in the spending either. So there's a lot of noise in the medical spending for a 0 to 1 year old child. And I'm not sure that the follow-up on sequencing is going to make this huge. Now, question on your mind might be, are these results actionable? That's a really hard question to answer because, again, as less presage so many of these issues, actionability is kind of in the mind of the beholder. You might distinguish if you wanted treatability, meaning there's a pill or a procedure, from actionability, meaning there's something you can do, including surveillance. But fortunately, we didn't have to define this. There is a ClinGen actionability scale, for better or for worse, that if it's considered actionable, it looks like a perfect diamond. If it's very non-actionable, it looks like that little triangle there. So let me show you all of the conditions in baby-seq 1. And keep in mind that the diamond would mean they were actionable. And sure enough, most of them approximate that diamond shape. And on that ClinGen actionability scale, all of them met criteria for actionability. So it's not like we were, at least in our first couple of hundred, it's not like we were defining things that were completely bizarre and non-actionable. These are real things that we found. We also found that when you find it in the baby, you define it in the family, right? So the top row is all the babies who carried the mutations, and the orange ones are the babies who were manifesting some sort of phenotype as defined by clinical exam, laboratory testing, or imaging. But none of the parents realized that something that they had was due to the same mutation. And some of them also had manifestations of the mutation that they were surprised to find. So if you're thinking about penetrance, at least in the short term, and we've already said penetrance really ought to be estimated in the long term, but if you're going to think about it in the short term, it's about 17% in the infants, 23% in the parents, but 35% in the family unit. So 11% of the babies have a mutation. 35% of those families are already manifesting undiagnosed genetic disease. And at least in our first iteration, all of them are actionable. Think about that. That's a lot of potential benefit. Now, we did something that pediatricians would mostly object to because we're a research project. We want to be on the cutting edge. We actually screen these children for adult onset conditions as well. And there is a feeling that you shouldn't do that because you're taking away the child's right not to know. And we respect that position. But in this research context, we wanted to understand what would happen if we did look for these things and if we did disclose them. And it was very clear in the IRB and in the consent, and the parents approved. In 3 of the babies, we found adult onset cancer predisposition. By coincidence, it happened that the carrier, the parent who was carrying that mutation, all 3 were mothers. And all 3 of those mothers in the 5 years since these disclosures have had risk-reducing surgeries. These mothers wanted to live longer to take care of these children. Surely this is a good thing. And I thought you might enjoy hearing a little bit from these parents. So let me step over here and see if I can make sure you can hear this. Okay. Let me.
You might have to enable it.
Yep.
Yeah.
Okay.
B. We just want our children to be healthy and happy.
The attraction of participating in the study was having that information and how empowering that information would be.
We just want our children to be healthy and happy. And if we can know that what could potentially be harmful to them, we can hopefully act on it.
When we got the results for my son, we had learned that he was a carrier of something called supravalvular aortic stenosis. You know, you think about those high school football players that just fall over and die in a football field because they had a heart condition that nobody knew about. So I was like, oh, well, but they had only known if there was some way to know. So I always kind of comforted myself by thinking, at least I know it.
The most critical thing that they found with my son is that he has the BRCA2 mutation. types of cancers, and that I should start to look into that.
Right.
I would rather my boys have me for longer than me get something that I know is a higher risk for me. Overall, just so happy that we have opted in, that we know our risks, that we're taking action steps to, you know.
I'm going to go ahead and skip over. So if I keep talking. Okay, there you go. Sorry about that. Okay, well, you get the flavor. Parents were appreciative. Those were not cherry picked, and in particular, one of the cancer moms said, "I'd rather be around longer for my kids." Now, we've expanded BabySeq into 7 recruitment sites, a much more diverse population, and we're finding the same kind of percentages in more diverse populations. And what you may not know is that this is really expanding around the world. So I formed with David Bick, the International Consortium on Newborn Sequencing 3 years ago. We had our first meeting in Boston with a steering committee that we invited the 8 groups in the world that we knew about who are working on this issue. And we have an executive director. And we've had 3 annual meetings, the first in Boston in 22, the second in London in 23, and we just got back from the 3rd one in New York City at the New York Academy of Medicine just a couple of weeks ago. And each one of these has been attended by more and more people. Each time, the last one between in-person and virtual is almost 500 people from 50 countries. So this is happening. You can see that there are sites all over the world that are now working on newborn and childhood sequencing. As it turns out, if you count China, there have been 68,000 children who have been sequenced as part of one or another of these studies. So this is a real thing. It's really happening. And but there isn't any commercial model. So a couple years ago, we started a public benefit for-profit company called Nurture Genomics. And this platform is up and running. We've been piloting it in 5 states: California, Texas, Illinois, New Jersey, and Florida. It's working very well. The friends and families who are trying it out are liking it a lot. And you can go online and get on a waiting list as we sort out extra funding for this. And if any of you are investors or know investors who might be moved to participate in this area, let us know, because we think we have to use the engine of commercialism to generate innovation, push the costs down, and make this available at scale. There are people trying to make it available at scale in the newborn screening ecosystem. And for a lot of reasons we can discuss, I don't personally think that's going to work. But it's important to try all of these different things. So here's our websites. Here's our social media accounts. Here's emails if you'd like to reach me. I'm happy to share these slides with you. And why don't we stop there? And I'd love to hear your thoughts and reactions to this and thinking about this for the future. Thank you.